Yes, good morning. First of all, thanks for the invitation. It's a big pleasure to be among this big guy today. I will try quickly, because the time is not so much, to give an overview of what we can do to isolate vesicles. I don't know how to move on. Oh, yeah, just here. Maybe. This one? Oh, okay. Never seen it. Okay, so a concept that has been expressed many times already, but I'm sure you will listen again and again about this, that we like to make our life complicated because extracellular vesicles are complex, so difficult to work with them, but we are doing a lot. You already saw this picture, beautiful picture about what we know so far. We know a lot since I started to work with extracellular vesicles. That actually is not so long time ago. I started in 2010. But I can tell you that, we did, or they did a lot. So now we know all these things. At that time, we just know exosome, microvesicles, apoptotic bodies. But the point is, as said already many, many times, unfortunately, we cannot isolate these vesicles yet, based on their biogenesis. What we can do is to try to look at the physical characteristic or biophysical characteristic of these vesicles. And this is what I'm going to try to show you today. This is a cartoon. But the reality is this. This is a cryo-electron microscopy of human ejaculate. You can easily see the sperm tail here. But the reality is this, that this is what we have in the biofluids. And I can guess that all of you agree that this is a mess, and we have to work with this. So, as I said already many times, we work in this context. So a lot of many things in the biofluid, and we have to dissect them and see if we are able to isolate vesicles, separating them from other things that we have in the biofluids. So the first message that I want to give you is that the EV preparation protocol is not a one-size-fits-all solution, unfortunately. It depends on the characteristic of your EV source. It depends if you are interested about EV subpopulation, maybe not, maybe yes, and also on the aim of your study. So, another important concept is that since our life is difficult, we also have to accept that EV isolation is always a compromise. So this is a picture that I got from an interesting review from Ann Hendricks, published in Nature Reviews Method Primers. And basically, we have to choose our bottles in few words. So, sometimes, again, depending on the aim of your project, on your idea, you just want to reduce the volume, the liquids of what you have, but you will be happy to keep everything that is there. Or maybe you want to reduce the volume, but also the other material that, in a bad way, we call contaminants, because they are not vesicles, but contaminants sounds really bad. Actually, it's not. Or it can be that we want to have a single EV subpopulation. So we want to try to isolate all those populations that I showed you in one of the first slides. It's obvious to know that in this sense we increase the specificity, but we also reduce the efficiency. I don't know what happened there. Anyway. We have several methods to try to do this. One of them is differential centrifugation. I'm sure that you know about this, but I will go a little bit more into details. Density gradient, size exclusion chromatography, filtration, and immunoaffinity capturing. Differential centrifugation, I guess that all of you know, but it's based on the size of vesicles. And again, since there is a huge overlap between the different EV subpopulation, we have to be careful, but it's very well used. So increasing the speed and the force and the timing, you will get always vesicles that are smaller and smaller. I don't know why it's going in this way. Anyway, the good point is that we can start from large volume. Basically, the limit is the tube that you can fit in the ultracentrifuge machine. And it will give you the possibility to concentrate the sample in a proper way. The problem is that I didn't put all this animation, but it's fine. You need the ultracentrifugation, you need the rotor, that I guess you know that they are expensive, and not all the labs have it. Since you concentrate the samples, at the same time, you also create aggregation. And at the same time, you also risk to have co-isolation with other soluble proteins. So in general, depends on the source of your samples, but the EV that you are going to isolate are quite dirty, we say. It's based on the separation, based on the size, but actually it's not perfect to separate vesicles depending on their size. Another way that I really like is to prepare the density gradient. It's based on the measurement of the tendency of a substance to float in some other substance. So in few words, you create several layers, as you can see here, with different density. You load your EV pellet, dissolved usually in PBS or other substance. You can load at the bottom or on the top of your tube. And then after quite long centrifugation, usually, your subpopulation of vesicles will be distributed between the layers. And this is what happened in the reality. I guess that you can see an enrichment of something here, but I can promise you that all the layers contain something, mostly vesicles. The good point is that you can separate vesicles from contaminants. You can isolate subpopulation of vesicles based on their different density. And you can also prepare it in a simple way. It means to prepare not all the layers, but just few of them. And in this case, we call it cushion. And it's mostly used to clean your pellet from contaminants. The problem is, again, that you need ultracentrifugation and rotors that are expensive. It takes time. And the volume is limited because you have to think about your tube. You have to create the layers that take space, of course, and then you have just maximum 3 ml for what I usually prepare. So your samples cannot be too large in volume. And keeping in mind that in this case you are just isolating the vesicles, then you suppose that you want to do something else. And the substance, the liquid where your vesicles will be, in this case in the density median, can have an effect on the further experiment. Size exclusion chromatography, again, is based on the isolation based on the size of vesicles. Basically, you have a matrix where your vesicles and other elements can flow through. And depending on the size, you will have first the large EV, and then the small EV, and then the protein contaminants. The good point is that it's mostly used to isolate vesicles from plasma, so you can eliminate, or you can clean your vesicles from the albumin, for example. You don't have the problem that you usually have with the density gradient, because in this case you can have PBS. So that is usually good to do other type of experiment. The problem is that it's not easy to isolate vesicles separated from other lipoproteins, for example, VLDL. You can prepare the columns by yourself, but it's not super, super easy. And your samples then will be really diluted. So keep in mind this. Another thing that we can do is to use the filtration. It's based on the use of some molecular weight cutoff. And usually how you perform this filtration, just using centrifugation. I don't know why it doesn't like this picture, but the good point is that it's easy to use, it's faster compared to ultracentrifugation and density gradient, but the problem is that you can lose some vesicles that will be stuck to the membrane, and since you use a force, with centrifugation of course, then we have the problem that eventually you can create the vesicles mechanically. Another very well used techniques is the affinity-based techniques. Is based on the interaction between proteins, mostly proteins that are on the EV membrane, and on their corresponding ligands, most of the time antibodies. It's good because it's fast, it's simple to use, and it has specificity if you know the marker that you are looking for on the vesicles, and this is a long discussion. The problem is that all of you, I guess, know that the ligands cost, the antibodies, for example, are quite expensive. You should be able to optimize some parameters, and it's quite well used to isolate vesicles from cell culture, so quite easy model, but it's more complicated when you want to use different sources of vesicles, for example, plasma. We have several of additional methods. I cannot go through all of them, also because I don't know all of them, to be honest, but I can tell you something. One of that is quite well used is the asymmetric flow field flow fractionation. Then we have the ion exchange-based techniques, the aptamers, the use of aptamers, and microfluid-based devices. You will know more about these microfluids, and there are also many, many others. So this asymmetric flow field flow fractionation, quite difficult name, so we call it AF4. Again, is based on the separation, looking at the size of vesicles. I'm not physics, so it's not easy for me. But for what I know, you have two plates separated by this permeable membrane at the bottom, and you have two forces, basic. The parabolic flow, in this sense, and the perpendicular cross flow. And thanks to these two forces, then you will have the separation of vesicles based on their sides. And differently from the size exclusion chromatography, in this case, you will have the small vesicles coming out before the large vesicles. The good point is that you can exchange the buffer, so it's quite important, mostly if you think about therapeutic application. You can have a lot of information about your vesicles, for example, size, distribution, morphology, and aggregation. But again, similar to size exclusion chromatography, then your samples will be quite well diluted. And for what I know, it's quite okay to be used at a small amount or small volume, but a little bit more tricky when you have larger volume. Quickly, ion exchange-based techniques is another way to separate vesicles on some physical properties that they have. And in this case, we look at the charge membrane. So we have the negative charged EV membrane and other elements that you put into the column that are positively charged. Aptamers it's a quite recent and really innovative technique. So preparing single-stranded DNA or RNA oligonucleotides, you can recognize something that is on the surface of the vesicles. Yeah, as example, one that I really like is these things that has been done by the group of Leiden. In this case, they used two EV isolation method So they isolated the vesicles positive to CD9, CD61 using immunoaffinity chromatography. And then they applied the asymmetric flow field fractionation. And they managed to isolate these new non-membranous nanoparticles that are called exomeres. They have been already mentioned. So why I'm saying this? Because until few years ago, it was impossible to think to isolate these small nanoparticles, non-membranous nanoparticles. So the message that I want to give you is that it's a quite difficult field. I know that all of you are struggling every second in the lab to isolate vesicles and have a good population of vesicles. But all together, we are doing a lot of things, and the things are improving. The method that I described to you are valid for other sorts of vesicles as well, like plants or bacterial or tissue. But the point is that, unfortunately, you cannot just copy and paste the method that other people use it. As Clotilde said, you have to optimize the protocol depending on your source. Also, between two different cell lines, something can change. Again, mentioned a lot of time, but this is really important and really useful. Follow the MySafe guidelines, because they can really help you, because they summarize what other people did, and they really help you to follow the, probably not easier, but the best possible way to isolate vesicles. I started with a quite negative message saying this is a mess, we don't know what to do. Actually, we know, but we have still work on that. But I want to tell you that the situation is improving, and it will improve all the time thanks to your work. And because, as I told you, when I started, basically, we only know something about exosomes, microvesicles, we call it at that time, and apoptotic bodies. Now we know that a lot of subpopulation of vesicles, membranes and non-membranes vesicles exist. And I'm sure that with the work from the entire community developing a new method, we will know always more and more. So I'm done, thank you. And if you want to know more, much more about what I said, here there are the references, and thank you.